Hello, I'm Jan Witkowski of Cold Spring Harbor Laboratory, and this is the occasion of the 89th Cold Spring Harbor Symposium on Quantitative Biology. And the topic this year is senescence and aging. And I'm delighted to have Ali Shilatifard with me. Ali, I've forgotten where you are.
I'm at Northwestern University Medicine in Chicago.
Okay. Yeah. Thank you.
Absolutely.
So I always think of you as being a regulation of transcription guy. What are you doing at a meeting on senescence and aging?
You're right on. So because a couple of my colleagues who are, see me in those meetings, this is the first question I asked when I sat right over there. What are you doing here, right? You're a transcription guy, you're an epigenetic guy, why are you here? And so they're correct. I'm a transcription and epigenetic person, and my lab studies regulation of gene expression, molecular mechanism of, and how this molecular mechanism implies into development of cancer, mostly, right, developmental disorder. And then once we find out what goes away in disease, can we develop targeted therapeutics to treat that? And that's what we've been doing for the past 30 years in the lab. And we have developed, basically the first almost 30 years ago, we showed that transcription elongation control is central for regulation of gene expression. Mm-hmm. This is around 1996, where basically here, majority of the colleagues, the leaders in the field, thought that initiation was it. Yeah. And once you release from initiation, who cares?
Just goes.
Exactly. And we have had the first link between an elongation control, a factor named 1119 lymphoid leukemia gene, as an RNA polymerase II elongation factor. And so ELL is a gene that translocates into another gene on chromosome 11 named MLL, mixed lineage leukemia. And MLL translocates to an array of chromosomes, and anytime you have these translocations, you have leukemia. Mm-hmm. And for the first time, we showed that the function for one of these translocation partners, ELL, is regulation of elongation of transcription. And the question became that what's the function of the rest of the partners? So we spent about 10, 12 years purifying all of these translocations from patients and asking what's within there. And what we learned at the end of the day is that although MLL translocates to many unrelated genes on different chromosomes, their gene products are component of the same complex, the super elongation complex, regulating the elongation stage. So that told us that elongation is central pathway for regulation of gene expression, and if you muck around with it, you can develop disease. And another disease that I'm the medical advisory board is CHOP syndrome, that basically the super elongation complex has a single point mutation, makes the elongation faster, and the kids have a lot of neurological developmental disorders, and so not only cancer, but development. So we asked the question that can we study these elongation factors instead of in a test tube, but inside the cell? Mm-hmm. So UKIOE in my lab developed this technology where you can do an AID, oxygen-inducible degron of these things, so you can degrade the protein specifically, and he started almost 8-9 years ago, very popular technique now. And he degrades them, and the moment the protein goes away, he asks what happens. Mm-hmm. And he identified series of this thing, and in a recent collaboration with my colleague at UCSF, Vijay Ramani, we started doing long-read sequencing, so we don't have to put contact together, where we can look at the entire transcript. Right. Mm-hmm. And we asked the question, what are the commonalities among all of these elongation factors we and others have characterized when you get rid of them at the moment of loss? And we learned that two factors, NELF and SPT6, have some link to senescence and aging. Right, some area that I don't study. And postdoc in the laboratory, Said Prost, was studying the role of these factors, and Said showed that actually you can get rid of NELF, cells will stop growing, add NELF back on, the cells pick back up going. Mm-hmm. And when he did sequencing for this thing, he found out that basically the SASP, this is the senescent-associated proteins, are actually expressed like crazy in the NELF. Then he tested SPT6, and same thing happened. So we asked the question, could be that basically NELF and SPT6 function somehow in the process of senescence. So the reason I'm in here is to learn more about the field, and I'm going to tell you what I've learned so far. I don't think we have a definition for senescence. This is my understanding of attending this thing for 4 days. But going back to the role of elongation factors and senescence, what we learn is that basically if you lose these two elongation factors, cell gets senesced, okay, and there's some problem, and they express all of this SAS protein to tell the surrounding that there's problem, take care of it. So I'm coming from the yeast genetic background, and in the yeast world, when we study things, we want to identify a function with the genetic screens, high copy suppressor screen or just a genetic suppressor screen. So we did the same thing in the loss of NELF or the loss of SPT6, the cell SENESCENCE. We asked, what gene do you knock out in the genome? They come back.
So this is yeast we're talking
This is
about.
no, this is all human.
This is
I'm just telling you my genetic
yeah,
background
okay.
is from
Right,
yeast, so
right.
we are treating human
I
cells
was
as with
wondering-
yeast.
I wasn't sure that yeast actually senesce.
So they don't.
Carry on.
No P16 or 21.
Right, yeah, carry on.
So basically, so in the human cells, we asked the question that if you reduce the level of NELF, you see senescence. What gene do you knock out in the genome that you come out of
compensate.
senescence?
Yeah, yeah.
And so say did that screen, bunch of hits. The top hit turned out to be a factor is an RNA polymerase II elongation factor named ELOA. So we said, does the SPT6 do the same thing? And UOA, a graduate student lab, did the screen, and she came up with the same thing. And when you overlap the two screens, there are 85% overlap on what the hits we get. So we asked the question, how does ELOA sort of suppresses the senescence function of the NELF loss? And we learned that upon NELF loss, there is anomalies forming at the 3' end of the transcript. And if you get rid of ELOA, those anomalies go away. So 3' end processing became very important. And I think 3' end processing defects sends a signal to the cells that we need to halt growth, and that's where the senescence starts.
So what's I must confess, I don't think I've ever thought about 3' processing. So what is the nature of that process?
We're still trying to figure it out. So what we see basically is we look at the movement of polymerase. Polymerase in the absence of NELF basically peaks up at the 3' and then go down, right? But if you delete the ELOA, that goes away and the cells can survive and they can move on. So as I said, I'm a transcription guy, epigenetic guy. We never worked on senescence. But from graduate school on, I knew that senescence defined as primary fibroblasts, you put them in culture, you can go 15, 20 cycles to stop growing. That's senescence. So we asked the question, when the fibroblasts go for 15, 16 cycles or 15 cycles, do you see this anomaly on the 3' end that we have seen? And the answer is yes. We see the same thing in the fibroblasts as they senesce. So we asked the question, what if you delete ELOA in the fibroblasts as passage 1? How many passages they go? They pass passage 16 and keep on going. And so we think that we have found something about the 3' end processing that causes senescence, and that ELOA plays an important role in that process.
So when you said they continue, how indefinitely have you
Well, we have
on
gone.
them?
I think Saeed has passed them out, 20 some odd cycles. We haven't gone further out. We got a lot of great questions from the colleagues at the meeting, the things that we need to be doing with that. The work's still not published.
And what cell type are you using?
We are using basically, these are the fibroblasts are human embryonic fibroblast.
Embryonic.
And then there's some, I think, early stage, fibroblasts from human. And we are testing a bunch of different fibroblasts. There are some fibroblasts that actually senesce, stage 60, passage 60.
Yeah.
Some are doing 20. We're going with the 20 ones because, we want to go to 60.
Sure.
And so we have tested multiple different fibroblasts, and we see the same thing. Both Saeed and Yo, a graduate student in the lab, they see the same thing. So now we want to do screens, we want to do analysis, and we have a model system to really go after that.
So what happens in cells that transform in culture? You've got a primary culture.
That's what I got out of this meeting. So now what I'm learning from here is that there are many ways of senescence. There's an oncogene-induced senescence, there is a replicative senescence, there is like an insult senescence. So now I'm going to go back in the lab and make all of those, right? And ask the question, what is the difference between all of these senescence type, right? And how does ELOA and transcription function in that process, right? And how can you solve the problem in here?
So do those various senescent types you've just described, they presumably have some commonality in terms of what's going on to the cell. I mean, apoptosis getting induced or...
So I think my understanding of senescence is that basically something happens in the cells. You've had DNA damaging agent or transcriptional defect or oncogene induced, and the cell actually stops growing to let the environment around it know that, I have problem.
Right.
And that's function of the SASPs to come in and solve that problem. That's what I got out of the meeting in here. Why do you do it and how do you respond? I'm not clear if I'm an answer. And my lab is more of a molecular biochemical pathway approach to this. So that's, I think, what I learned is that there's a long list of experiments that we would like to do from what I learned through the meeting. So for me, actually, I was talking to lunch with a colleague. I go to a lot of epigenetic meetings and I go a lot of to transcription meetings. And most of the stories I've heard before, I'm reviewing them. So I don't get anything new out of that. But here, I feel like I'm a graduate student. I love it. I'm taking notes. I've got experiments to do. And I really suggest for people who are listening to this, go to the areas, meetings that you don't work on, and a lot of ideas will come from it.
Yeah. So, dare I go a bit political and ask how your funding is at the moment? And
for us, we still fund it. I think Northwestern has some issues with that. The funding needs to be solved, and I think our administration is working with the government trying to see how we can solve that problem. I think funding for science is very important. Science should be a political factor. When people come to our hospital, we don't ask them any question about their politics. Our job is to treat, right? Our job as a scientist is to discover, and our job as professor is to educate broadly. And I think that's I hope is our goal that we are moving forward. And I hope that funding would be there, but I think basic research funding is fundamental to the livelihood of this country. And if you look at the dollar for dollar, how much money has been put into research and how much the economy and health has benefited from it. Yeah. And I don't think we have to go far from the COVID vaccine. A lot of people think the COVID vaccine was developed in the year by pharma. Not really. There were 30 years of experimentation on DNA, recombinant DNA technology, how to clone things, how to express things, nanotechnology, how to get things inside the cell. All of that was put together and we formed a vaccine that was very helpful. But none of that happened really by pharma. It happened at academic level. Yeah. And I think all of that probably was funded by National Institute of Health and National Cancer Institute and similar governmental funding from other countries.
Yeah. You mentioned one of the functions is education, and clearly you've been educated here at this meeting.
Absolutely.
When did you first come to a Cold Spring Harbor meeting, Ali?
The very first Cold Spring Harbor meeting was the Symposium in 1998.
Oh.
Was on transcription, right? And I was just started my faculty position, and we just had discovered ELL, right? So I had a poster, and I came in here and I met very good number of colleagues like Danny Reinberg, Shelley Berger, Ramin Shekatar, Tony Kuzurides. I met all of those folks here at that symposium and we became a lifelong friends. Yeah. And I think that's the beauty of the meetings, in-person meetings here at Cold Spring Harbor, that not only you educate yourself, you form lifelong bond with colleagues that you have similar interest in life. Yeah.
Well, I think that's a very good point to stop.
Absolutely.
Thanks very much, Ali.